So I'm going to talk about I should have to do my disclosure. One time Jim Heath gave a 10-minute presentation of his disclosures. So I'm going to talk about theranotics. Probably most of you are not familiar with this, relatively new. So it combines diagnostics and therapeutics. It uses the same molecule, a small peptide or a small molecule or an antibody, to either image and phenotype in patients, and then with a different isotope to radioablate cells. So it shares some of the good things about drugs without some of the limitations, and some of the good things about external beam radiation therapy. 50% of all patients cancer patients get external beam radiation therapy, so it's accepted and effective. So the way it works is we'll label, and I'm going to focus on small peptides today, but we'll label the peptide with F18 or gallium. They're positron emitters, so we can image the peptide and assay it and do the kinetics in the body. Or we'll switch the label on that same peptide to either lutetium-177 or actinium-225. They're long-lived, either beta particle or alpha particle emitters. So they'll radioablate the cells that the peptide is bound to. So with lutetium, about 90% of the radiation exposure is within about 0.5 millimeter. Actinium, which is alpha particle, 90% is within 20 microns. Now, as we go through this, these peptides not only bind to proteins on the cell surface, but very quickly, within about an hour, 95% of them are internalized. So even further, moving the radiation to the interior of the cell. So first we'll look at neuroendocrine tumors. So we pick a target, a somatostatin receptor in this case, and there are 100 to 100-fold increase on the surface of neuroendocrine tumors. Now, when you look at the images that I show you here, and as I go through and change the cancer, the target, and the probe, I don't want you to just think about detecting cancer. They do that. But more importantly, they will phenotype the cancer cells throughout the entire body of the patient and determine whether the patient has the protein target for the therapy, treat them, and if they don't treat them. So you see on the left a patient with neuroendocrine tumor with multiple tumors in the liver. So the question is, does the patient have the protein target? The answer is yes. You treat the patient with lutetium-177 dotatate, and dotatate is the small peptide. And then you do the follow-up exam, imaging exam, and ask the question, have you ablated cells? You can clearly see the cells are ablated. Now, this is a trial that was carried out on theranotics called the Neder Trial and published in the New England Journal of Medicine to evaluate the either with or without a chemotherapy agent, octreotide. And the protocol is done in a way that the patient receives the lutetium dotatate first, and then later is followed up with the chemotherapy. You can see the impressive results in terms of progression-free survival and overall survival. And also in the New England Journal of Medicine paper, it kind of parenthetically said that the side effects were mild and manageable. Now, that is not a trivial issue. And with all of the theranostics so far that I'll go through, the side effects in all of them are mild and manageable, and I'll show you why. So, change the target, change the probe. Now we go to prostate cancer. So the target is PSMA, and a small peptide with very high, all these peptides have very high affinity and specificity for the target. The previous neuroendocrine tumors, if you notice on the bottom left of the slide, very quickly went through FDA approval and reimbursement. Now I'm going to come back to that and also explain why. For the gallium PMSA, the NDA was submitted in March, and a projected approval time, the NDA for the therapeutic, is in 2020. So again, we go to the image, ask the question, does the patient have the protein target? The answer in this patient is yes, with a large array of metastases throughout the bone from castration-resistant prostate cancer. Treat the patient with lutetium PMSA 617, go back and image the patient, have you ablate the cells? And again, you see in this patient, the answer is yes. Now we're going to switch from lutetium, the beta particle emitter, to actinium, the alpha particle emitter, which has much more dense exposure to it. To the left, again, you see a patient with a horrible distribution of metastases throughout the bone of prostate cancer, and these patients have failed all other treatments. You treat, and the patient's PSA is 3,000. You give the patient 3 treatments, re-image them. You see that now many of the cells have been ablated, and the PSA is dropped to 0.3. Give the patient one more treatment, the PSA drops to 0.1. The images show that you have ablated the cells. Just to the right, another example of that with very large tumor burden. The PSA in this patient is 419. Treat the patient with 2 doses of the actinium PMSA. PMSA drops to 3.5. One more treatment drops again to less than 0.1. So these treatments are amazingly effective and also have mild and manageable side effects. Now, one has to go through FDA approval, has to go through reimbursement, but you also have to go through medical adoption. You have to educate medicine about the diagnostic or therapeutics that you introduce. Now, another prostate cancer patient sent to UCLA, but it's noted it's an outside urologist, not one of ours. So our urologists are very familiar with the Theranotics approach. They refer their patients to us. They believe in it. They understand it. And in fact, we have a 4-month wait time because the growth has been so quick that it's expanded beyond our capability to supply the service. So on the left, you see a CT at the lungs and also down to prostate bed. The CTs are normal. And then you see the superimposition of the gallium PMSA assay on top of the CT, and you see a lesion in the rib, and also one in the prostate bed. Now, the outside urologist, unfamiliar with PMSA, decided that the rib lesion was an artifact. Now, the patient also did report that he fell and had rib pain. So between that and normal CT, he decided not to treat the rib lesion. He did treat the lesion in the prostate bed, even though the CT was also normal there, but he had a prostatectomy and recurring PSA. Now, like all the PET scans that examine the biology of disease, they delineate malignancies by the biological processes as opposed to the ambiguities that exist with CT and MR. Now, 9 months later, we see this patient with the 2D projection images. Now his PSA has gone from 1.9 to 19. You see that small rib lesion is now large in multiple metastases. So this was a mistake, but these kind of mistakes are going to be made early on. So now we are treating the patient with the lutetium PSMA. So There's a company in France called Advanced Accelerator Applications. It's a pet radiopharmacy company. And they acquired the intellectual property for the neuroendocrine diagnostic and therapeutic and received FDA approval for the diagnostic in 2016. Now, the diagnostic approvals are a lot easier and quicker than the therapeutics because the PET imaging probes are generally in FDA under what's called generally safe and effective. So they actually go fairly quick, small number of patients. And in 60 million clinical PET studies, there are not one reported complication. Now, interestingly, only 23 months after the pre-meeting with the FDA, the diagnostic was reimbursed rather quickly. CMS began their approval, reimbursement in June 2016. Novartis acquired AAA in October 2017 for $3.9 billion, and the FDA approved the therapeutic in January 2018. So all of these dates move quickly. Indocyte is a medium-sized biopharmaceutical company in Indiana. They licensed the lutetium PSMA 617 from a German company, ABX. By July 2018, Indocyte stock rose 600%, producing a market cap of the smaller company of $1.2 billion. FDA allowed Indocyte to start their trial at phase 3. Now, the reason they allowed them to start at phase 3, which is true of all the other theranostics, is that in Germany, in particular the University of Heidelberg, but multiple universities, they actually go through the clinical trials using indigent patients, and they pay for them themselves. So when we, in our country, submit an IND application, we already know it works. We already have published trial data from Europe. That's why FDA will let us start early. Now, Indocyte was considered in 2018 as one of the top 3 biotech stocks, and then was acquired by Novartis for $2.1 billion. Now, to many other companies, the growth in theranostics has been quite remarkable, both academic and commercially. So the other companies, at least that we know are in theranostics now, are J&J and Sophie Biosciences, a company founded by Jim, me, and others. Indy Molecular, that involves ISB and Jim. Bayer, Progenics, Astellas, and so forth. But there are actually other pharmaceutical companies that we know privately are also moving into theranostics. Now, part of that reason will become apparent a little bit later. So, again, we're going to change the target, change the probe. So this time we're going to focus on fibroblast activation protein, FAP, that's bound to cancer-associated fibroblasts. And these CAFs are unique to cancer cells. They're not detectable in normal tissue throughout the body. Now, the latter part is important also in terms of side effects. And I'm about to focus on that issue a little bit more. So when you look at solid tumors, in some ways you can consider them as pseudo-organ systems. They are composed of cancer cells and many other normal cells that they corrupt to serve the purpose of the tumor. But they also build a barrier in the stroma to keep the immune system out of the tumor, and also, of course, a barrier to penetration of drugs. So the goal in this diagnostics is basically blow up the tumor. So 2 objectives are going to radioablate cancer cells like we showed before, but then break down the stroma. And Now, there have been many drug companies that have been trying to break down the stroma for penetration and immune access, but that's not been very successful to date. So the probe is actually a small molecule inhibitor of FAP with very high affinity and very high affinity and selectivity for FAP. And if you look at the kinetics, now with PET, there's something that we can do. We can do in patients, do all the kinetics to watch the probe or a drug as it enters the body and where it goes throughout the body over time. Now, kinetics is less popular than it used to be, but remains still important. So I want you to look at these images. The images I've shown you before and the ones in the last I'm going to show you in a minute, I know you're not used to reading these images, but there are only two things I want you to look at. So you look at this breast cancer patient with a FAPI1 probe targeting FAP, and you see in 10 minutes already there's enormous concentration of the probe in the tumors, and also that the probe is cleared out of the nonspecific tissue in terms of side effects in 10 minutes. And in kinetics, these probes are also internalized very quickly. 95% of the probe and protein are internalized in about 60 minutes. Now, when you look at the images, that's the biodistribution of the probe. And remember that same probe is used to image and treat. So when you look at the images, you know the biodistribution for the therapeutic. Does it hit the target? Is it retained in the target? And how quickly does it clear from nonspecific tissue? The imaging tells you that. It tells you whether the patient has the protein target, for which you will treat them, or you will not if they don't. Now, these images are actually from the University of Heidelberg, and I'm going to come back to a program that a lot of us are involved in, but they have very quickly, remember, they can go into patients very quickly. So they have already done gallium FAPI studies in all those cancers shown to the right. And these are the images of them, broadly across many different cancers. But again, you look at the images the way I mentioned for the therapeutic. Is it hitting the cancer? Is it clearing away from the soft tissue? You see in all these different cancers, that is true. Now, whether this is effective in all these cancers like you saw before, whether it really does blow up the stroma and expose the tumor to the immune system and to drugs, we don't know. So those therapeutic trials have now been started in Germany, and we'll just have to wait and see how that goes. So this all takes place in academic industry partnership, UCLA, UCSF, ISB, University of Heidelberg. So how many universities in America were founded in 1386? That's amazing. It's the oldest university in Germany, but my God. So at Heidelberg, much like Lee has been intuitively and instinctively the master of many things, there's an MD Ph.D. that heads and developed most of the modern theranotics, who's the master of this. And he knows things beyond the principle. There are fundamental principles in theranotics, but he knows things that other people don't know. So he very quickly goes through the trials. He doesn't waste his time on irrelevant things, but focuses on the right things and makes all this work. Just simply amazing. His name is Haberkorn. He's an MD, PhD. He actually has a relatively small lab, and he wants it to be small. Not everything is about being big. In fact, small is where the best of everything comes, even all of you, and ISB as well. So we have two companies, Sophie, that Jim and I are founders of, and Indy Molecular that Jim and Lee founded, that also work together. And we work together in an environment where we move the solution to problems anywhere on this slide. Whoever can solve the problem best, that's who takes that portion of the problem on. But we all work together. The faculty own these companies. They're their companies, of course, along with the officers and the investors, but it's their company. Now, this is a very unusual slide. I have become unpopular many times in my life, and this is another way. But I've always believed, like Lee Hood has believed, that diagnostics are really the key to therapeutics. Measurements, measurements, measurement. So I did a very simple signal-to-noise analysis to try and put the focus on the diagnostic as you approach the therapeutic trial. Now, signal-to-noise, a very formal approach, is the power function. So I asked a very simple question. What are the relative number of patients required in therapy trials for a given outcome as you change the percent responders? So signal to noise, signal responders, noise non-responders, do the analysis. Now, under reasonable assumptions, the answer is if you have 20% responders, which is pretty normal for clinical trial, drug trials, it takes 100 patients. If you had a way to increase the responders to 40%, it takes 25 patients. If you had a way to further increase it to 80%, it takes 7 on a relative scale. Now that results in corresponding reduction in cost, time, and number of patients exposed to the risk of therapies that don't serve the purpose of the trial. This is a dramatic way to impact not only therapy trials, but our use of treatments for patients. But you have to develop new classes of molecular diagnostics that can achieve these enrichments. Theranotics actually does that. It is not a theoretical example, it is a real example. We have the diagnostic to select a patient with very high response rates and eliminate those who don't have the target. Now, the last part is a surprise. Well, there are two surprises. So, this is a PET-CT scanner. It was developed by my previous student, Simon Cherry, who's at the University of California, Davis. Now, he submitted a request to build a total body PET scanner that could examine all of the body simultaneously, do the kinetic interactions between all cells throughout the body. In Lee's world of systems biology and yours, this is total systems biology throughout the body. When Brown and Goldstein won the Nobel Prize for cardiovascular disease, they gave a lecture and said, if you want to improve cardiovascular disease, work on the liver, LDL. But we know all of these systems are connected, but we've never had access to them before. So this system can handle a 6'5" tall patient and examine the kinetics throughout all cells. Now, a funny thing happened. After years and years of failing with NIH, the reviewer said it's not possible to do this. There are many hardware, many mathematical, many computational innovations required to make this happen. And they kept saying no. But Simon kept persisting. And finally, they gave him a little bit of money to do a Monte Carlo simulation. And with that, they gave him they turned it down again, gave him some money eventually to build some of the hardware components that people challenged whether they could really do that. But over the years, a company called United Imaging in China had been recruiting Simon's students and mine back to China. And they're the largest supplier of X-ray, ultrasound, CT, MRI in China. They became a very impressive company. I think surprising to a lot of people, they have technology that equals or exceeds GE, Siemens, and Philips. And they're a very proud company. So Simon convinced them that they could build this Explorer, a total body PET CT scanner. And then he said to the CEO and the head of R&D, which they had agreed to do this, don't you want to know the market forecasts? And they said, no, we will build it and find out. Now, we used to do that in our country. So they did, they built it. Now, that's what it looks like. So, to your left is a GE, Siemens, Philips type modern PET scanner of what are called whole body PET scanners. To the right shows you this large array of detectors with enormous innovation to make all that work. And if you look at, so in positron imaging, positrons are annihilated with electrons, E equals mc squared. 2 photons are emitted back to back and detected by opposing detectors. Those are called lines of response. And we collect them around the patient, do a little math, reconstruct the images. So in modern whole body PET scanners, there are about 0.4 billion lines of response collecting data simultaneously. In the Explorer, there are 92 billion. 92 billion. So it's over 50 times as efficient as the modern system, but covering the whole body. Now, in the articles below, Simon and his colleagues propose increasing it to 100-fold, 200-fold. Now, the 100-fold, they can do. And it's going to take some innovation and some invention, but they can do it. 200-fold will probably require some miracles, but Simon believes in miracles, like all of you do. So in whole body imaging conventionally today, we typically take 20 minutes to do the whole body. To the right, you see a 20-minute total body image with Explorer. This is the probe fluorodeoxyglucose, so it looks at glycolysis throughout all the cells in the body. But you see to the left, in 18 seconds, they can come very close to that, or in a minute. So they have amazing efficiency and temporal sampling to do the kinetics of biochemistry and biology and pharmacology. At the right, you see a cross-section down to the liver, and you see the aorta, but it's the glucose metabolism of the wall of the aorta. Now, to the right, I'm going to show you a whole body kinetic study. You're going to inject FDG in a foot vein, and you're going to watch that probe distribute throughout the body. That's going to move very quickly, and you'll see the times at the top. So it's coming up through the vein, into the lungs, in the heart, into the lungs, back in the heart, moving throughout the body. It's now in the heart, the liver, the kidneys, moving into the brain. So this builds a new platform technology where one can do the kinetics of biochemistry and biology and pharmacology throughout all the cells of the body. The two greatest things that change science are instruments that you can see and measure things you never could before, and theories that you consolidate the knowledge to the point you can explain it and even predict things you didn't know about. So, this is the last slide. So, I collect cars, and turbo Porsches are really fast. And so this 70-year-old man buys a turbo Porsche. He takes it out on the highway, driving along at 50 miles an hour, and he thinks, this is a turbo Porsche. Takes up to 80, at which point a highway patrolman takes after him. But again, he thinks, this is a turbo Porsche, man. Takes up to 120. And then he thinks, what the hell am I doing? I'm 70 years old, trying to outrun a highway patrolman, cranks it down, and pulls it over. The patrolman comes up to the car and says, look, it's Friday, 7 o'clock. I'm about to go off duty. If you can give me an excuse I've never heard before, I'll let you go. So the old man said, well, a year ago, my wife ran away with a highway patrolman, and I thought you were bringing her back. And he said, have a nice weekend, sir. And that's it. Thank you. I think I went over my time. Oh.
Thank you for a great presentation. I was wondering, are the scanners that you just showed used for prevention or just diagnosis, or how do you imagine them being deployed? And is there any deployment yet, and are you willing to share any cost?
Well, we share everything. You tell me a secret and it's going to go on the web, man. So, we live in an open world, and now, I have to tell you, I boxed for 11 years, and boxers know it hurts when you get hit in the ears, so I can't hear very well. So what did she ask me?
Sorry about that. I'll repeat it. Is that better?
Yeah.
Okay. So first of all, thank you for a great presentation. Secondly, I wanted to know if the instrument is intended for prevention and scanning or just diagnosis and treatment, and is it deployed, and are you willing to share any pricing?
Yeah. So all those things. So there are pathways to go from preclinical science to clinical research to trial to patient care. So we treat patients that use neuroendocrine tumors and prostate cancer. Now, neuroendocrine tumors are reimbursed, approved, and everything. The prostate therapeutic is not approved yet. So we went to the FDA and said, we want to take care of our patients. Our patients demand it. And we want to provide the service. So they allowed us to do cost recovery. Even, we're running the trials, but we're allowed to take care of patients on cost recovery. The patients have to pay, but they eagerly pay. We're building a new Theranotics center, but everything has been moving so fast, both academically and commercially, we're having trouble responding to that, to increase quick enough. Even our recruitments of new faculty in translational theranostics are hard because there's so many jobs for them. Now, we are trying to diversify. Jim has developed a high throughput synthesis screening small peptide technology. Our goal at UCLA is to move to ovarian, pancreatic, lung cancers, preclinically, and then move it back up that pathway. But remember, the work is coming from that whole group of people, from the German universities, from UCLA, from San Francisco, and from our companies. And there are companies to serve what we believe in. What are you doing?
I had a question.
I thought we did that last night.
I know, but I forgot the answer.
Yeah, I know why you forgot.
So I think, as you know, but I think a lot of people don't fully understand, one of the challenges in the types of theranostics and probes is that every probe has to be made very, very quickly.
Right.
And the chemistry...
For the diagnostic.
From the diagnostics. And probably the therapeutics is a little more forgiving, but it's still on a day basis, not an hour basis anymore.
Yeah.
And so I wonder if you could talk a little bit about what the technology limitations are in terms of democratizing these various theranostic probes.
Yeah. So one of them is called fluorine. So the imaging I showed you was done with gallium-68, which is a positron emitter, but it's expensive and it's very limited supply. The worldwide PET radiopharmacies are all based on fluorine-18 diagnostics, but fluorine-18 has 2-hour half-life. And fluorine is in 2 chemical forms, F2 explosive, F minus. You're asking chemists to do chemistry with inert gases. I mean, it's non-reactive. So very clever techniques have been developed to do very rapid end-stage fluorination. But the critical issue is get the target right, and then be clever enough to produce and screen the peptide, the small molecule, for the properties you want. Bind quickly to the target, be internalized, and very quickly clear out the non-specific tissue. Now, there are no principles about non-specific tissue. You're either lucky or you're not, but these peptides have been amazing. But remember that with the imaging part, we can very quickly do the biodistribution in patients to say, no, that doesn't work, or it does, and then move forward.